Hi, welcome. I'm Professor Bryan Ballif. I'm a professor in the biology department and one of the neuroscience faculty members. I'm glad you're considering, as a prospective student, the opportunity to work at the University of Vermont in your graduate program. As you understand, all aspects of neuroscience involve genes, and those genes encode proteins. And those proteins enable the processes of biology and neuroscience to function. And when those processes go awry, we get diseases of the nervous system. So I'm sure you understand that it's important then to analyze proteins and be able to understand their function and what they do, and what proteins interact with our proteins of interest, and how those proteins function inside of a neuron or inside of a cell. The Proteomics Core is a wonderful facility that enables us to analyze proteins. And to analyze proteins, we need to understand what they are. So you might, in your work, purify a protein, and you might want to know what it is. But if you run it on a gel and have it stained, you won't understand what that protein is. You won't actually know its name until you figure out it's a linear sequence of amino acids. So we have special mass spectrometers, high-end mass spectrometers, purchased by the National Institutes of Health through a grant to the University of Vermont, that enables us to analyze proteins and to determine their identity. So the mass spectrometers are really high-end, high-precision instruments. They enable us to determine mass values with precision down to about 0.002 to 0.003 of a Dalton. That's not very much mass, and that enables us to be very precise and very accurate in our measurements, and enable us to give you the high quality data that you need to accomplish your work. We also have proteomics courses, ones that I teach, that you could take, and that would enable you to further understand protein analysis by mass spectrometry. I've been a mentor to one of the NGP graduate students, Riley St. Clair, who's currently postdoc at the University of British Columbia. Riley did analyses in the Proteomics Core to help her understand how proteins during development play critical roles in neurons during pathfinding. Riley did fantastic work, published her work, and was able to move on to a wonderful postdoctoral position. The size of my laboratory has on average 2 to 3 graduate students. This enables personalized attention so that we can get students and give students the training that they need to succeed, and also create a very friendly, collaborative, warm environment in order to conduct studies in a way that is fun and in a way that gives them the community that they need to succeed. We also have several undergraduates in the lab, and for those of you interested in training undergraduates, you'll have an abundance of opportunity to do so. The faculty members in the Neuroscience Graduate Program are highly collaborative. My research is collaborative with Dr. Alicia Ebert in particular, and we have grants from the National Science Foundation to enable us to conduct the research in neuronal positioning during development that is so important to a basic fundamental understanding of neuroscience. When you join the University of Vermont's NGP graduate program, you'll be joining a community. And the communities are collectives of individual labs that come together for activities, as well as, for example, apple picking or making banana bread in the lab, or any number of different type of activities that we do as a group.
My name is Todd Clason. I work in the basement of the Given building, and the reason I work down there is because that's a best place for the microscopes that I have. And I run a facility. It's a live imaging facility, and it's part of the Vermont Center for Cardiovascular and Brain Health. So the equipment that we have in the basement, we have 3 main modalities that you might be interested in. One of them is a spinning disc confocal microscope. And the idea there is that that kind of piece of equipment goes really fast. So if you have an item that has calcium signaling or some other neurological event that is sub-second and you really want to see what's going on, that piece of equipment does an amazing job to do that kind of work. Now, you might be someone who's more interested in structure, and you might have a cleared brain, and you're looking at some sort of construction in the brain, neural network kind of a thing. You've labeled something green and something red, and you want to see how everything is knit together. And in that case, you may use the confocal microscope, which makes absolutely gorgeous images, 3D images, very provocative, very nice kinds of things to get someone's attention if you have a poster or anything else. And if you're really pressing the envelope, you can use the multiphoton microscope, and that system allows you to look deeply into live tissue. I'm there again to help you figure out what the most appropriate technology is. Another nice thing about the University of Vermont is that the cores are integrated so that if you have some idea that is an imaging idea and you really want to know if you can do it, and the equipment that I have is not quite what you need, there's a whole nother facility that I work closely with as part of a larger core. It's called the Microscopy Imaging Center. And they have electron microscopes, both transmission and scanning electron microscopes. And they have a very nice laser confocal scope, which is sort of a flagship model, which is a little bit more complex than the one that we have in the basement. They have atomic force microscopy, and they also have super resolution microscopy. And if you're really, really interested in microscopy and more than just one single modality, we have a whole course that we teach. Doug Tachi's, Dr. Doug is a co-teacher in the course that I run, Neurological Sciences. It's NSCI 328, so it's Techniques in Microscopy, and it's a great overview, especially if you intend to set up your own lab and you want to know some of the tricks of the trade about what kinds of things to look for and how to buy the right digital camera for your microscope and what kind of objective should you get. So those kinds of things are something that you might be interested in. The ability to actually learn some of the techniques and do them yourself is kind of rare. And I think that for graduate students who want to have are aware of the fact that, part of the job besides getting the degree is building a skill set, so that it's very rare, frankly, that you're going to do a degree and get a PhD and immediately get a job that is exactly that thing that you wrote for your thesis. It's unlikely. But during the thesis process, you learn so much that if you're absorbing that and you're really building those skills, then you're quite marketable in that way. So I see students that do all sorts of things. They may stay in higher education, but they may go off and do something else. And there's lots of technological fields that are looking for PhDs. And microscopy is certainly a big one for that. The thing about microscopy these days is that many new technologies are coming online, and it's fun, and so that's why I recommend doing it. Actually, it's the fun part. There's a lot of difficult work that needs to be done as a graduate student, a lot of repetition and things like that, but there's nothing as exciting as doing some complex labeling experiment and then being actually able to see that on a microscope. So yeah, it comes highly recommended.
So I'm Cal Thomas. I'm in Dr. Green's lab in the Department of Psychological Sciences, and I work in Dewey Hall. In my second year, I joined my dissertation lab, and we decided that we wanted to be able to do experiments using DREADS manipulation. And in that manipulation, we needed to be able to do high quality imaging of the neural tissue because we were looking at expression of immunofluorescent compounds like green fluorescent protein and M-cherry. And so I actually took a microscopy imaging course with Todd Clason and Doug Tachi's, who are directors of the imaging cores, the MIC and the COBRE Neuroimaging Core. And so I learned a lot about imaging and how to use microscopes and stuff like that in that course. And then in the subsequent semester, I ran my first experiment using the DREADS manipulation. Being able to use that equipment, or it was the C2 fluorescent microscope in the COBRE Neuroimaging Core, was really critical to our ability to even kind of carry out those experiments, because we're a relatively small lab. We don't have, multi-million-dollar microscopes to use on our own. So it was really critical for us to be able to get that imaging done at something like the core here. And I think that was really one of the biggest benefits of kind of UVM in general, is that I could be in this relatively small lab. Currently, I'm the only grad student in the lab, and I'm still able to do these kind of state-of-the-art experiments using really novel techniques, because we have access to really high-quality machinery or microscopes through the cores.
Hi, my name is Thomm Buttolph. I'm the research technician in the COBRE Cellular Molecular Core Facility, and welcome to the lab. So the core director is Dr. Sheryl White. She is in charge of overseeing this space. She's extremely helpful for project design, data analysis strategies. She has a vast experience in molecular biology techniques, so she is definitely the go-to person should you have any questions regarding your experimental design and setup. I personally take care of a lot of the protein and nucleic acid work. In addition to that, I specialize in CAD design of brain implants. In the facility, we have a lot of high-tech equipment that would be extremely expensive for an individual lab to own. So this facility is basically providing you both the equipment and the expertise in order to be able to further your research. We have multiple cryostats available for cryosectioning, and actually Dr. White has been working on developing a technique called spatial transcriptomics, which starts at the cryostat collecting tissue, and then we'll probe that tissue for various gene expression. We have a LICOR Odyssey infrared imager at our disposal for doing Western blot analysis. It allows you to realize two targets, two different wavelengths at the same time. Our lab is fortunate to have access to two liquid handling robots. We have a QIAGEN QIAcube and a QIAGEN QIAgility. We have a complete tissue culture facility with biosafety cabinet and CO2 incubators. We also have a Countess cell counter, which is your equivalent of a digital hemocytometer, which makes plating out cells a lot more consistent. We have a Biotech Synergy H4 multi-mode plate reader. It's primarily used for protein assays and ELISAs, but also comes with two injector modules should you choose to do some type of experiment where you'd like to inject something and immediately read the effect of that injection into your multi-well plate. Fairly recently, we've added a resin-based 3D printer system in addition to a plastic FFF or FDM type printer, 3D printer. Both Dr. White and I are here to help you troubleshoot. Even if you're not actively using the core equipment, we're still here if you need to diagnose an issue that's giving you problems. We do have an open door policy should you need to come in and talk about an issue with your research that's really causing you a whole lot of grief. Come and talk to us. That's what we're here for. We are the problem solvers. So don't hesitate.
Hi, I'm Megan Perkins. I'm a 6th-year neuroscience graduate student in Margaret Vizard's lab. So we have used the Cell and Molecular Core Facility for a lot of different experiments. We've used multiple types of their centrifuges. We have many centrifuges in our lab, but they're on the smaller side. So we've used their larger centrifuges for large test tubes, as well as centrifuges that can spin plates for bacterial experiments or qPCR experiments. We've used their cryostats, which allows us to section frozen tissue and then use that tissue for immunohistochemistry experiments in our lab. We've used the nanodrop, which allows us to assess the concentration of the RNA in our samples before we use those samples for any further experiments like qPCR. And we've also used their quantitative PCR systems to look at gene expression in different types of samples. So we've used the cell and molecular core facility for a ton of different experiments, and we're super, super lucky and grateful to have all of these fantastic instruments just at our use. Sheryl is the manager of the core facility, and she is a great manager. She's always around making sure things run smoothly, super willing to help with anything that any quick questions, anything that you would need help with. Thomm is the technician. He's a fantastic technician. He knows these machines inside and out better than any of us do here. And he also ensures that everyone is trained on each of these machines and trained properly. So Thomm has been super helpful with tons of my experiments. There's been multiple times where I've forgotten a key piece for the cryostating instruments, and he's been there to help me find whatever materials that I need. He's always there when I forget one crucial step for a program to say, like, don't forget about this button. And so that is really helpful. So they're always super helpful with any questions that we have, and it's great that they are always around the facility in case we need that.
I hope that you enjoyed the virtual tour, and we will see you in the fall.